it will be about alternative splicing, regulation and deregulation of alternative splicing that has very far-reaching consequences, could have very far-reaching consequences. And one example of what alternative splicing deregulation can do is the splicing of this RON oncogene. RON oncogene is RON is a tyrosine kinase receptor, as many other receptors. Is a tyrosine kinase receptor for is a receptor for a certain class of proteins that are called macrophage stimulating proteins and members of the scatter factor receptors family. So what happens when this receptor is, tyrosine kinase is activated by the ligand? What happens is that it promotes cell dissociation, motility, and invasion of extracellular matrices. So cells become more motile, and can invade intracellular and can move around. Okay? So it promotes, it's involved in what is called invasive growth or scattering. And this process, which is the EMT, epithelial to mesenchymal transition. Epithelial cells are not very mobile, they stay there in clusters. The mesenchymal cells move around, form new organs, and so on. It's very important, of course, it's a cell plasticity program, which is important for development, embryogenesis, and other things. But unfortunately, it's very important also for cancer development. So cancers can stay there or become metastatic because some of their cells undergo this epithelial to mesenchymal transition. That's why this guy is an oncogene. He's an oncogene because some friend of ours in Turin many years ago discovered that certain colon carcinomas contained very high level of this oncogene, or better, very high level of a truncated form of this oncogene. The receptor is an heterodimer which is derived from a precursor which is cut by protease, so you have two subunits. What they've observed that certain tumors, colon carcinomas, contained an abnormal, a very high level of a form of this receptor that was deleted of a very tiny exon, 49 amino acids in the beta chain. But the deletion of this very tiny exon had a profound effect because it removes the proteolytic site that splits the precursor into alpha and beta, and so you have you don't have the processing, and as a result, this form called delta RON is constitutively expressed also in the absence of ligand. You don't need any macrophage stimulating factor to have the activity of this. So in this sense, it's an oncogene. When it is mutated or overexpressed and so on, it promotes some form either of cancer or metastatizing cancer and so on. So this friend of ours, the cell, asked us to investigate what was the mechanism that caused the skipping of this exon and that had such profound consequences on the activity of this oncogene. And we did a lot of work and discovered that delta-RON, in fact, confers invasive phenotype to the recipient cells. So if you transfect this DNA, this gene, into cells, they become, and you can measure that and you can observe that. And in fact, this form is overexpressed in colon and breast cancers. So after we did a lot of work, of course, and we discovered that the choice between this kind of splicing, in which the exon 11 is included, and the other one in which the exon 11 is excluded, is determined by two sequences which are present in the downstream exon, exon 12. And the downstream, you have an enhancer of splicing, you have already heard about it, and the silencer of splicing. You have a cassette of regulation. In the case of delta-RON, what happens is that the splicing factor SF2, which is an essential splicing factor, very important in the regulation of alternative splicing, also called ASF or SF2, binds to the enhancer, makes this acceptor site of splicing much stronger than it's in its absence, and so the splicing apparatus, the spliceosome, skips and this exon is excluded. Okay? So this is a typical mechanism. So the key factor is SF2, because exon 12 contains a binding site very good for SF2. We determined that. And the problem is that to switch from inclusion of exon 11 to exclusion of exon 11, it all depends on the level of SF2. But what is very scary is that the level of SF2 which are necessary to switch are two-fold increase, 2.5, threefold. You don't need a lot of increase of SF2 to switch from one splicing to the other, and to switch from one type of cells, epithelial, to mesenchymal cells. And these cells are completely different. So an increase of twofold of splicing factor 2 through the action on this RON oncogene, this receptor, cause a very big change. For example, mesenchymal type cells are spindle shaped, while epithelial cells are cobblestone, they are all stuck to each other. And there are markers of these two types of cells. For example, beta-catenin is normally nuclear, it redistributes in the cytoplasm. The cytoskeleton modifies, so actin distribution is modified, and E-cadherin, that is a typical marker of aggregation, the level of E-cadherin decreases a lot. Okay, you can see here. And these cells, you can measure, there is a test where you can measure the capacity of the cells to migrate through extracellular matrices or synthetic porous membranes. So you measure how many cells you find on the other side of the membrane, and you see that a cell line in which we have cloned ASF, SF2, and we have double level of ASF, SF2, are much more proficient in migration compared to the control. So this is very interesting because this process, EMT, is what happens in tumors. It turns out that in metastatizing tumors, talking about solid carcinomas of epithelial cells, not leukemia, the central part of the tumor is sort of epithelial, and the cells at the border are of the mesenchymal type. And in fact, what are the markers of this transition? The marker of this transition is that you have a decrease in E-cadherin and a reaccumulation of beta-catenin in the nucleus, and an increase in these are transcription factors, and they have other features. So, but the message is that these cells at the border of the, unfortunately, they are able to migrate, and they do migrate. And when they establish a metastasis in another part of the body, the process is reversed. So you go from this type to this type in the new position. So it's very tricky. Turns out that there is a very simple model for reproducing the EMT transition. And is to take these colon carcinoma cells, culture, and grow them at high density or low density. So these cells at high density, are of the epithelial type. Cells at low density are of the mesenchymal type. In fact, you can see there is no E-cadherin at low density. Is an association marker. And for example, beta-catenin, which is normal in the cytoplasm, move to the nucleus. These are just hallmarks of this transformation. So, and what is the proof? What is the demonstration that this is a good model? This high and low density. That cell density controls the splicing profile of RON and the expression level of SF2. You see, at high cell density, you have the inclusion of the exon. At low cell density, you have the exclusion of the exon, and you have almost all delta RON. Okay? So at low cell density, and you see the increase in SF2-SF is not much, 3 times, something like that. But that's enough to switch completely the splicing program and to cause the transformation in the properties of the cells. Now, so SF2 is a key factor in regulating the splicing of this oncogene, which is important for the scattering property from the invasiveness of the cells and so on. But what regulates SF2? And this is very interesting. And you will hear about it more from Lynn Marquardt, probably. It turns out that these splicing factors, many splicing factors of the SR type, so there is a family of splicing factors to which SF2 belongs, have in the 3' non-coding region an intron, which normally is not removed, is included, okay? Because when it is removed, it causes the so-called, it causes the fact that this stop codon is recognized as a nonsense stop codon because it is the distance of the exon-intron junction is more than 50 nucleotides. So when this intron is removed, the resulting mRNA, which the coding region is the same, just is the 3' non-coding, is degraded by the NMD system. Okay? And so we believe that the regulation of the level of SF2, and remember the level of SF2 is very finely tuned and regulates the splicing of RON, for example. We believe that the regulation of SF2 is not through the promoters that are switched on and off. Is regulated through the inclusion or exclusion of these tiny introns in the 3' non-coding and region. And this is true. And in fact, we observed that cell density, for example, regulates SF2 alternative splicing. So at low cell density, when cells are of the mesenchymal type, they migrate. Why they migrate? Because they have more full-length isoform, and so they make more SF2, because only the full-length isoform is translated into the protein. The one which is destroyed, degraded by this mechanism. In other words, and you see at low cell density, you have more SF2 compared to high cell density. But you see the differences are not much. No? 2-fold, 3-fold, 2.5, so on. And we checked that, you know, this was not an artifact. That in fact, so the phenomenon was due to the nonsense-mediated degradation and not to some kind of inhibition of protein synthesis, A-specific, okay? But I don't have time to explain all these experiments, but the essence is that, yes, SF2 is regulated in this way through the homeostasis between two forms, the one that contains the intron and is full length and is translated and becomes protein, and the one in which the intron is spliced that is degraded through this pathway of degradation that you will hear a lot about tomorrow by Lynn McWatt. Okay, so SF2, if you look, in fact, if you look in tumor samples, these are real tumor samples, and you look at the ratio between the full length and the NMD isoform, you see that in most samples, there are 14 samples, in most samples, you have a very high level of full length, so you have a high expression of SF2. So SF2 is also an oncogene. When it is overexpressed, it causes tumors or metastasizing tumor. And this was observed also in a Kreiner lab in Cold Spring Harbor. And you see, you have, but the ratio is 2-3 times higher. So the level of an increase of 2-3 times of this oncogene, it's enough to make a mess. Okay? Now the story becomes even more interesting because then we asked, nonsense-mediated decay might play a role in the expression of the oncogene in tumors. Okay, that's probably. I skip this part that shows that. So I said that if you grow cells at high density or low density, you have different behavior. Well, it turns out that you can reproduce the same result. For example, if you use low density cells and you can add to low density cells conditioned medium from high density cell, for example. Conditioned medium means that you take high density cells, you wash them from the medium, and you resuspend in medium without serum. And so what they do, they secrete all possible things, growth factors, cytokines, and so on. This is called conditioned medium. Conditioned medium can produce the same effect as the cell culture itself. That means that what causes the effect is something soluble that is present in the medium and is secreted by the cells. And we will see what it can be. But I want to go ahead. In fact, this shows that, if you have low density cells and you add conditioned medium from high density cells, you see beta catenin goes into the cytoplasm. So you have the markers of EMT activated, and the shift in delta ARon splicing. So this fact implies that there is a signal transduction cascade which is involved in the message that transmit the message to, and to make a long story short, the signal transduction pathway that we discovered is involved in the regulation of SF2 is the ERK1/2 kinase. And so the secreted factors that control this alternative splicing of SF2, they act through the RAF, MEK, ERK1. This is a major signal transduction pathway. And these are the evidence that, in fact, the phosphorylation pattern, but I don't want to go too much into that, because you can find it on the paper. It's on your CD, all the evidence. And so we ask, well, but what goes on in the 3' UTR? What controls this splicing that controls the homeostasis of SF2? Okay. And we looked at the 3' UTR of SF2, and what happens is that it is known that at the border of the intron there are two ultra-conserved elements. But by ultra-conserved it means 100 nucleotides, 100% conserved between man and mouse, for example. 100 nucleotides, 100% conserved. Next to it, there are two binding sites, very good consensus, for this protein, SAM68. SAM68 is an RNA binding protein, very important. The splicing regulator is called SAM68 because SAM stands for Sarc activated during mitosis. That's the way it was isolated. SAM68, and this is very strange because these two sites are very conserved, but what is conserved is the distance between this site and the border of the intron, all the ultra-conserved elements. So it's a very important region, and probably the interaction with SAM68 is important. In fact, as I said, SAM68 is an RNA binding protein. These are RNA binding domain. And this phosphoryl is a target of that kinase, very important signal transduction kinase, ERK, is known to be a target, and is phosphorylated into here, this amino acid. Now, in fact, at low cell density, SAM68 is phosphorylated. Now, the first thing we did in collaboration with Emanuele Buratti, who is that guy in the audience, we demonstrated that, or they demonstrated, that in fact SAM68 binds to these two sites, right? It binds, and if you mutate the two sites, this is the site. If you mutate, it doesn't bind anymore. So it's a real specific binding. And then they tested that by producing a mini gene that contains this region, and then transfect it into the cell. You reproduce on the transcript of the mini gene the same thing that happens to SF2 non-coding. Okay, so what happens is that SAM68 controls the non-mediated decay of the ASF, SF2 mRNA, and cells that overproduce SAM68, like this, see, they are spindle-shaped, and they have the features of mesenchymal cells, while those that don't overexpress SAM68 are epithelial, cobblestone cells. And what is interesting is that SAM68 is so strong in determining this phenotype that in this clone that overexpresses SAM68, you never go back, you never go reverse from mesenchymal to epithelial cells. So SAM68 controls the nuclear-mediated decay also of endogenous, not only on the mini genes, but also of endogenous. And to make a long story short, if you look at SAM68 level in the tumors, in those 14 tumors that I showed you before, those 14 samples, you see there is a good correlation between the level of SAM68 and the ratio between SF2 full length, therefore protein, and SF2 nuclear-mediated decay. So there are exceptions. For example, here you have a lot, not much SAM68, but a lot of this protein. But because, you know, the action of SAM68 is due either to the level of SAM68 or to the phosphorylation state. So SAM68 maybe is not much, but maybe is very phosphorylated in this case, and so it induces. Anyway. So the key factor in the end of all this hierarchy of factors, the key factor is this RNA binding protein, which is the target of the ERK1/2 kinase that by regulating the inclusion or the exclusion of a small intron in the 3' UTR of SF2, determines the level of the SF2 protein, okay, which is an oncogene. And the level of the SF2 protein impacts on the splicing of RON, which is something that determines whether cells become able to move around, to cross the barriers, and so on. And all these events are due to very small changes in the levels of this factor. Twofold, threefold. And this tells how delicate equilibrium a living organism is. Okay? It's enough that you shift by twofold, threefold level something, that you have a lot of consequences, some good and other bad. Okay, and I hope 5 more minutes.
5?
Okay. Now, this delta RON splicing, aberrant splicing, indicates that delta RON is a possible target for drugs. So if you can find drugs or some treatment that reverse the splicing and includes this small intron into the protein, then you don't go to the mesenchymal type cells that migrate, make metastasis, and so on. And in fact, there are at least three approaches that are being tested, some in our laboratory, other in other places. And there are also some preclinical trials going on. And I briefly summarize what one possibility is to use morpholino antisense oligonucleotide. Morpholino oligonucleotides, they are modified oligonucleotides. They don't have the usual ribose. They have another molecule in order to prevent degradation. You know, when in the cells you have an antisense oligonucleotide, the RNase H degrades everything. Well, the morpholino, they directed towards the SF2 binding site. So you block this site. The idea is you block this site with the nucleotide, and therefore you don't have this splicing anymore. You include the SF2. And in fact, the first results are encouraging. If you look at in the presence of morpholino oligonucleotide, you have a switch from delta RON to RON. Okay. And it's specific because this, for example, caspase 9 splice. Caspase is a protease, and is a target of SF2. The splicing of a certain intron in this protein requires SF2. Is not affected by this morpholino oligo. So the morpholino oligo recognizes the sequence which is present only in RON. Another approach is more interesting. It's called targeted oligonucleotide enhancers of splicing strategy. So what are these? Instead of having blocking this site, you have an oligonucleotide bipartite which has a region that hybridizes to the exon, and then it has a tail that binds, is designed to bind splicing factor, in this case SF2. And also this oligonucleotide, this approach works in the sense that you have a switch of the RON splicing while you don't have an effect. This is the caspase and the two other genes that are affected by acid. This oligo, of course, this is modified in order not to be degraded by the RNase H. And the third approach, which is based on the use of small molecules that hit the SF2 activity directly, so the splicing activity. And these are indole derivatives. They are being tested, several indole derivatives, and some of them are promising in the sense that they change. You see, this is the normal delta ron. Some of these derivatives, for example, the first three, they do change the pattern, while they are not active on these other genes. So they are being tested. So that's essentially what I wanted to tell. The people who did this work, most of all, Claudia Ghigna and Giuseppe Biamonti, in collaboration with some people here, and other people around Italy and Europe. Yeah. Thank you very much.